Hello, my name is Kelsey Brown, and I'm going to tell you a bit about my research here at the University of Arkansas. The title of this research is named Building a Better Drug. But first, let's start with why I do this work. Fungus exists as part of our body's natural fauna. However, part of that fauna is known as Candida species. When these species are allowed to grow out of control, it can cause illnesses such as oral thrush or systemic infections in vital organs. Current treatments are polyenes, azoles, and echinocandins. Polyenes have limited uses because of high toxicity to our own cells. Some species have shown a natural resistance to azoles, and other species have developed resistance to both azoles and echinocandins over time. Our bodies have our own ways to ensure that we stay healthy. In our saliva are proteins called histatins. These proteins have both antimicrobial and antifungal activity. Of those histatins, histatin 5 has the most prevalent antifungal activity. Therefore, our lab has derived several small peptides from histatin 5 in order to develop a safer and more efficient drug. These small peptides have shown a greater increase in antifungal activity than histatin 5. However, we still don't understand the killing mechanisms employed by these peptides. The first step in uncovering how our peptides manage to kill infectious fungal species is to determine how it enters the cell. I will be adjusting the membrane fluidity of the cell to investigate this. In order to adjust the membrane fluidity, I will be treating cells with DMSO, which can cause the membrane to become more rigid. Other cells will be treated with benzyl alcohol, which causes an increase in membrane fluidity. We hypothesize that these fluctuations in membrane fluidity will affect the peptide's ability to kill the fungal species. The next step in our work would be to look at the membrane components that could be assisting the peptide in crossing the membrane. In order to look at these components, I will be using mutants from the Saccharomyces cerevisiae library to look at changes in resistance in our various mutants. Other members of our lab will be investigating the killing properties of the peptide once in the cell. There are poor treatment options available. They are either too toxic or not efficient. The goal of this project is to understand the killing mechanism of peptides derived from histatin 5 by starting at the cellular membrane. Long-term goals of this project will be to develop a safer and more efficient drug.